scientist at Rosen Center for Advanced Computing at Purdue University, and along with me, Haley Carter at Northwestern University's Research Computing and Data Services, we organize this seminar series. Today's seminar is being recorded, and we'll be posting this on our website afterwards. So even if your colleagues who couldn't attend this series, please let them know, and they can catch up with this talk later on. A few quick housekeeping notes. Please keep yourself muted during the talk. We'll take questions at the end. You can raise your hand or using the Zoom reaction, or just drop your questions in the chat and we'll read them out. The seminar will run 35 to 45 minutes, and afterwards we'll have the Q&A. With that, let me introduce today's speaker, Iris Liu. Our speaker today, Iris Liu, is PhD candidate in Driscoll Graduate Program at Northwestern University's Feinberg School of Medicine. Iris works in the lab of Dr. Benjamin Singer in the Division of Pulmonary and Critical Care Medicine, where her dissertation focuses on developing novel enzyme-based methods for DNA methylation profiling and applying them to identify predictive markers in respiratory disease. Before her PhD, Iris completed a master's in neurobiology at Northwestern, where she worked on CAR T-cell migration in glioblastoma models, and her undergraduate work at Bryan Moore College focused on axonal development. She brings a broad benched computation perspective to her current work. Iris has published in journals including Nucleic Acid Research, the Journal of Clinical Investigation, and Science Advances. She's also a recipient of the Kugel Fellowship and serves as a genomic compute cluster consultant and bioinformatics workshop instructor at Northwestern. She's very much in our world of HPC and genomics. Today, Iris will present her work on RREM-seq, a reduced representation enzymatic methylation sequencing method that enables single nucleotide resolution methylation profiling from ultra-low DNA inputs. How she has applied to identify methylation signatures associated with severe COVID-19 pneumonia outcomes. With that, Iris, floor is yours.
Thanks, Arun, for the wonderful.
You are cutting off. We heard your first few words, and then it got cut off.
What about now? I'm thinking it might be my headphone. Okay, sounds good. Let me start sharing my screen. Okay. Okay, everything looks okay? Okay.
Yes.
Okay. Yeah, thanks again, Arun, for the wonderful introductions and offering me the chance to sharing our work of developing DNA methylation sequencing methods, which allow us to perform DNA methylation profiling from clinical samples from patients with severe pneumonia. So here is a brief outline of what I'm planning to talk about today. I will start with the scientific background that motivated my projects, including our clinical context, severe pneumonia, and how Tregs is involved in regulating lung tissue repair. I will also briefly discuss the technical challenges currently in the field of DNA methylation profiling and how that shape my methodological approach. From there, I will walk through my experimental studies and results. Okay. Pneumonia, including SARS-CoV-2 pneumonia, has a massive and ongoing disease burden. This map on the left here breaks down global SARS-CoV-2 cases by WHO regions, with a larger circle indicates a higher case count. On the right, we can see how staggering these numbers are. SARS-CoV-2 alone has caused over 7 million deaths worldwide, with more than 700 million documented cases up to date. Influenza, though more familiar, continues to kill between 290 to 650,000 people each year, with annual US cases ranging from 9 to 35 million. Taken together, these numbers really highlight the urgent need for a better mechanistic understanding of viral pneumonia. In my lab, we are specifically interested in regulatory T cells, which is a specialized subset of CD4 T cells. In both human and mice, Treg would accumulate and expand in the alveolar space in response to injury. There are growing evidence suggesting that Tregs are involved in many different important functions following acute lung injury in mouse models. During acute respiratory infection, effective pathogen clearance usually require a very robust inflammation, but resolution of inflammation also the restoration of immune homeostasis are essential for the following tissue repair. Treg cells has been shown to inhibit other T cell population, including CD4 and CD8 T cell and B cell responses. And this process would prevent excessive inflammation. Tregs could also interact with alveolar macrophages, and this would promote neutrophil clearing for a process called lung resolution. Besides this more traditional immunosuppressive role, Treg cells are also actively engaging in tissue protection as well. They could limit collagen deposition and also steering the macrophage populations towards a more pro-repair and anti-inflammatory phenotypes. In addition to tissue protection, Treg is also actively engaging in tissue repair. They could produce many different types of growth factors, including the most famous one called amphiregulin or ARAG. Treg productions of ARAG has been shown to be required for type 2 alveolar epithelial regeneration, and that's a classic hallmark for lung repair. Okay. So during their development in the thymus, Treg acquire a very distinct epigenetic architecture that stabilizes lineage commitment and is required for maintaining all those functions I mentioned in the previous slide. Genome organizer SEBI1 is first recruited very early during Treg development to the Treg super enhancer sites. These regions are associated with future Treg identity genes, including the master transcription factor FOXP3. Working with other chromatin modifiers, SEBI1 would opens up the chromatin and organizes long-range contacts between promoters and enhancers. This step would then prime the super enhancer regions for TET enzyme recruitment. TET enzymes then catalyze the demethylation within these regions that includes loci encoding for FOXP3 and other lineage-defining genes like CTLA4 and IL-2 receptor alpha. In addition to this demethylation within the Treg super enhancer regions, we and others have shown that regulatory regions around effector genes. So these are the genes that are more inflammatory and have an opposite effect compared to Tregs. These regulatory regions need to stay methylated in order to suppress their expression for Treg to maintain their lineage identity and function as well. So it turns out the methylation patterns within these Treg super enhancer regions are fairly conserved. In the study that first characterized the super enhancers, the author shows using mouse splenic T cells that Treg cells are hypomethylated, so it has a lower level of methylation profile compared to conventional T cells. And using human mature alveolar T cells, we also observe the exact same patterns as well. Again, Treg cells seems to be hypomethylated compared to conventional T cells within these regions. Okay. Our group is part of the Successful Clinical Response in Pneumonia Therapy, or SCRIPT, System Biology Center, and that gives us real access to alveolar T cell samples from critical ill patients. These patients are all intubated, and a procedure called bronchoalveolar lavage, or BAL, is performed as part of their routine care in order to identify the source of suspected pneumonia. During these procedures, saline is instilled into the infected lung in order to sample thousands of alveoli and then draw out. We then use a portion of each of these BAL samples to sort immune cell populations. That includes CD4 T cells. All patients would usually have their first BAL within 48 hours following intubations, and BAL was performed throughout the course of mechanical ventilation. From these sorted populations, we generated multi-omics data sets in order to understand how this immune cell behaved in the injured lung. Okay. In the recently published SCRIPT study led by my friend and colleague, Dr. Nick Markov, we show that BAL samples from patients, specifically from patients with severe SARS-CoV pneumonia, they show an enrichment for T cell populations that includes CD4, CD8, and also Treg cells as well. And this seems to be a SARS-CoV-2 specific effect because we didn't observe this enrichment in patients with other diagnoses. That includes patients with non-pneumonia respiratory failures, bacterial pneumonia, and also other pneumonia as well. We then stratify all of our patients based on their clinical outcomes, so either discharged from the ICU or diseased. And we found out that also specifically in patients with severe SARS-CoV-2 pneumonia who later recover, they seem to show an enrichment for Treg cell proportions. And this effect was not found in patients with other diagnoses as well. Okay. So based on these previous studies, we know that Treg cells is involved in many essential functions following lung injury. Methylation is a key regulator for Treg cell function, and patients with severe SARS-CoV pneumonia who later recover seems to have a higher proportion of Treg cells. All of this leads to our current hypothesis that different sources of respiratory failures might be associated with distinct Treg cell associated methylation signatures, especially those related to the Treg superenhancer as well, superenhancer regions. To answer this question, we propose to profile alveolar T cells from critical ill patients with severe SARS-CoV pneumonia, bacterial pneumonia, and non-pneumonia respiratory failure as well. In mammalian genome, DNA methylation is most commonly found in CpG dinucleotide, that's cytosine followed by guanine. The most important task for any technology to profile DNA methylation would be to distinguish methylated cytosine from unmethylated cytosine. Currently, there are many different methods to transform unmethylated cytosine in order to distinguish them. The most well-used one is called the bisulfite-based conversion. It's still considered the current gold standards. It could convert unmethylated cytosine to uracil and later sequence them as thymine. And this is a one-step chemical reaction. Bisulfite conversion is great. They're very cheap and easy to use, but has a huge caveat. This chemical reaction is very harsh on DNA samples. Usually, more than 90% of the DNA will be degraded after the treatment. So it wouldn't be a good fit for low-input samples, for example, our clinical samples. To address this limitation, team from New England Biolab, they propose an enzyme-based alternative. So instead of using a chemical reactions, they could achieve the same methylated cytosine to uracil conversion using a two enzyme-based step. Because this process is 100% enzyme-based and there is no chemical reaction involved, it's very gentle on the DNA and could usually preserve the majority of the samples. Okay. So this cytosine conversion technologies can be applied either genome-wide or to an enriched subset of CpG-dense regions. The whole genome methylation sequencing, as the name suggests, it profiles methylation across the genomes. In mammalian genomes, CG dinucleotides or CpG, they show a very non-uniform distribution and mostly cluster in CG-rich CpG islands and other regulatory regions. So sampling throughout the genome is a comprehensive but not very efficient way to profile methylation. Alternatively, reduced representation methylation sequencing, or RMS, they enriches the DNA samples for CG-rich regions prior to the conversion step. This enrichment step would greatly enhances sample throughput and reduce its sequencing cost because we would not longer need that many reads per samples. And this would usually allow us to profile methylation on a relatively large sample size and also enhance statistical power for all of our downstream analysis as well. When we first started this project, one of the biggest challenge we found is the number of Treg cells we are working with. So for each BAL samples, we can usually sort out around 1,000 cells on a good day, but we also have a lot of samples with only 600 to 800 Treg cells. After DNA extraction, that roughly translates to around 1 nanogram of DNA, and that's a teeny tiny amount for any sequencing library generations. And when we first started the projects, there is no available technology in the market that could handle and input this low. To address this challenge, we developed a novel workflow specifically optimized for low-input clinical samples. We call it REM-seq, or Reduced Representation Enzymatic Methylation Sequencing. We show that REM-seq could successfully profile samples with less than 1 nanogram of input and publish this work with Nucleic Acid Research in 2025. REM-seq starts with enriching CpG-rich genomic regions using restrictive enzyme digestion, specific MSPI. After that, we use the enzyme-based technology to convert methylated cytosine to uracil. And then we use a protocol optimized specifically for low-input libraries to perform random priming, adaptive ligation, amplification, and indexing before we sequence them as non-directional libraries. Okay. We first tested and optimized our REM-seq protocols using input ranging from 2 to 25 nanogram. We picked 25 nanogram because that's an input level that the current gold standard in the field, RBS, works pretty well. To assess the library size distribution and qualities, we use a platform called tape stations. In these traces, the peak represents a population of library fragments, and the x-axis over here plots for base pair, so that's the library size. And the y-axis over here plots fluorescence intensity, and that's an indication for final library concentration. At 25 nanogram RBS, that's reduced representation bisulfite sequencing, it successfully generates good quality libraries. But once we reduce the input to 2 nanogram, RBS starts to show a lot of issues. First of all, the y-axis over here, there's a huge difference in terms of the scale. So it's actually a whole magnitude of difference. So this library over here is tiny. It's very small amount. And all of these small peaks here, it represents primer dimers. So we have a lot of primer contamination in these RBS libraries. In contrast, we found out that our REM-seq workflow could successfully generate good quality libraries using both 25 and 2 nanogram of input. We then titrate down the REM-seq input to be below 1 nanogram. We use 1, 0.5, 0.1, and 0.05 nanogram of DNA to generate the REM-seq libraries, and they all looks pretty good. Well, we start to see a little bit of primer contamination in the 0.05 condition, and the library distribution gets a little bit wider, but we would still consider this good quality library for sequencing. After confirming that our workflow would give us good quality libraries, we then designed the experiment to really benchmark REM-seq performance. In this experiment, we use conventional T cells and Treg cells as a system to benchmark REM-seq performance because we know that these two cell types has an established methylation difference. So it will be a test for REM-seq accuracy. We included 3 reduced representation groups in this experiment. That would be RBS, the current gold standard in the field that we're benchmarking against, and REM-seq with 2 different input levels. We also included 3 whole genome groups as well. So that's the counterpart of what we have in the reduced representation groups. That's the whole genome bisulfite sequencing and 2 whole genome EM-seq with 2 different input level. And again, we only included the low input in the enzyme-based methods because bisulfite-based methods could not handle input level that low. Okay, we defined a standard input as 25 nanogram, a low input as 2 nanogram. And for each of the groups, we included 5 conventional T cells and 5 Treg cells. So in totals, we sequenced 60 samples to benchmark our REM-seq performance. Okay. After we generated and sequenced all of our libraries, we first look at 2 important QC metrics. The first one is library molarity. That's an indicator for the amount of sequenceable molecules each workflow generates. At a standard input of 25 nanogram, we found that all 4 workflow would give us more than enough DNA molecules to pull for sequencing. But we do have a little bit of issues with the low input whole genome EM-seq workflow. We have to increase the final PCR amplification cycles quite a bit in order to get enough libraries for us to sequence. But in contrast, we have to see that REM-seq has no problem at all with generating enough libraries using this small amount of DNA input. Another important QC metrics for DNA methylation library is the cytosine conversion rate, or sometimes is referred to as cytosine conversion efficiency. All of our workflow incorporates cytosine conversion as part of the library generation process. And so having an efficient conversion is really important because all the downstream analysis would assume the remaining cytosines in the libraries are all methylated. Having an efficient conversion would significantly compromise the accuracy of the downstream methylation calling. To test for that, we included lambda bacteriophage DNA in all of our libraries. And by mapping our reads to the phage DNA, we will be able to because this phage DNA is 100% unmethylated, so by perform our methylation callings using this phage DNA reads, we'll be able to estimate a cytosine conversion rate. In all of our samples, we observe lambda phage CpG methylation around 0.5%, and that would correspond to almost 100% cytosine conversion efficiency. Okay, so next we move to compare CpG coverage in a global level. So that's across the whole genome at 1x, 5x, and 10x coverage threshold. So this analysis would only include CpGs with at least 1, 5, and 10 reads. I know this is a gigantic panel. How we structured it is the 3 whole genome groups is always on the left, and the 3 reduced representation groups is always on the right. And they're in the order of bisulfite-based methods, followed by the enzyme-based methods with 2 different input level. So at 1x minimum coverage, so that's without any filtering based on the coverage, standard input RBS and standard input REM-seq here, they detected a similar number of CPGs, while low input REM-seq, showing light blue color over here, they detected slightly less. But once we increase the minimum coverage threshold to 5x, we no longer see this difference. And same goes with 10x coverage threshold as well. To further compare the performance of REM-seq against the current gold standard RBS, we also compare the within-group CPG concordance between these two methods. Concordance analysis is a test for consistency, so it measures how consistently methylation score at each CPG site agree across samples, and that's within the replicates in the same groups. In these panels, each number represents the average concordance score between a given pair of samples. For each CPG site, we defined a concordant call between two samples as having an absolute difference in beta values of less than 0.15. Well, within groups, concordance vary quite a lot between RBS replicates. Standard input and low input REM-seq both show superior and more consistent concordance rate. We also calculate the Pearson correlation coefficient between a given pair of samples in the same groups at 10x minimum coverage. In these panels, the lower left here, that's the pairwise methylation values. And then the upper right corners over here shows the Pearson correlation coefficients. Similar to the concordance analysis, REM-seq show very consistent within groups CPG profiling. And both standard input and low input REM-seq samples, they have a Pearson correlation coefficient of higher than 0.95 in any given pair of samples. So the previous two analyses I talked about is a test for consistency, but another important matrix for sequencing library is how accurate they are. To test for that, we use the same concordance analysis, but instead of comparing within groups, we are comparing RBS and REM-seq libraries against WGBS. In this analysis, we're treating the WGBS libraries kind of as a ground truth because it has the most comprehensive coverage. So similar to the within groups comparisons, our REM-seq libraries also demonstrated superior between group concordance when compared to RBS, highlighting that RBS would provide a more accurate methylation calling. Okay, so after assessing REM-seq accuracy and consistency, we also want to compare its coverage within the CG-rich regions because that's the main advantage of using a reduced representation sequencing methods. At 1x coverage, we've found that within the CPG island regions, that's a very CG-rich genomic regions. RBS and standard input REM-seq and low input REM-seq, they detected a similar number of CPGs, while the REM-seq RBS samples over here varied quite a lot. But once we increase the coverage threshold to 5x, the two REM-seq groups starts to outperform RBS. And when if we increase the coverage threshold to 10x, the two REM-seq group not only outperform RBS, but also outperform the two whole genome EM-seq group as well. We believe that really highlight the REM-seq superior performance in profiling this CG-rich regions because for all of the whole genome libraries, we sequence them with at least 6 times more depths, but REM-seq will still give you a superior performance. Okay, yeah, we repeat the same analysis in the promoters regions and also observe the exact same trend. REM-seq starts to outperform RBS at 5x and it really shines at 10x coverage threshold. Okay. In the previous analysis, we calculated the number of covered CPGs in CG-rich regions. And here we plotted the average coverage. So that's how many times each CPGs were covered. And that would be a demonstration of how even our coverage are. In these regions, both standard input and low input REM-seq outperform whole genome EM-seq and RBS. On average, REM-seq would detect around 10 times more CPG calls compared to whole genome EM-seq and around 4 times more CPG calls compared to RBS. Okay. Next, we move to validate REM-seq performance in identifying methylation differences between cell types. So that's the conventional T cells and Treg cells we use in this analysis. We filter the reads to look for those mapped to the Treg super enhancer regions and plot out the methylation values. These panels on the figures on the left, on the y-axis, we're plotting the methylation value, so that's the beta value, against their cumulative proportions on the y-axis over here. In this plot, a leftward shift would usually indicate hypomethylation, so that's lower methylation values. And you can also see in our box and Wixter plot here, showing that Treg cells are hypomethylated compared to conventional T cells. Based on these results, both standard input and low input REM-seq successfully detected the established biological difference in methylation status between the two cell types we use in this experiment. Okay. So all the previous data I talk about are generated using mouse splenic T cells. So what about REM-seq performance when using real clinical samples? To test for that, we designed a pilot run using 7 paired conventional T cells and Treg cell samples from patients with severe SARS-CoV pneumonia and use them to generate REM-seq libraries. We confirmed that these libraries show a very similar performance in library concentration and cytosine conversion rate compared to the mouse data. And we also observed no significant correlation between sample DNA input and final library concentration as well. Okay. To compare the methylation status within the Treg super enhancer regions in these samples, we mapped the regulatory elements from the original MM9 mouse reference genome onto the HG38 human reference genome. And the Treg super enhancer methylation status trend are still consistent with our previous data, showing that Treg cells are hypomethylated compared to conventional T cells. Okay. So after this pilot run, we are really confident about REM-seq performance when profiling our low-input clinical samples. We then moved to design our study. For this study, we want the BAL T cells from early clinical time points, so ideally within 48 hours following the intubation. We reasoned that potential biomarkers from early clinical time point would be the most informative for later clinical decision making. We also want to prioritize samples that we already have the bulk RNA-seq datasets, so we could integrate transcriptional and methylation datasets together. That give us 77 samples in total. And again, we have 3 clinical groups. That's patients with non-pneumonia respiratory failures, bacterial pneumonia, and SARS-CoV-2 pneumonia. We generated and sequenced all of these libraries, and they all look great. Okay. After we receiving all the sequencing results, we did some quick QC check. On average, the cytosine conversion efficiency of these libraries is about 99.5%. So that's awesome quality. And we have around 80 million reads per sample. Currently, we are actively working on understanding these REM-seq datasets, and here are some sneak peek from our exploratory analysis. We first assessed the global CpG methylation status measured by our EM-seq. All patients' cohorts show a similar global CpG distribution, with the majority of CpGs clustering near either unmethylated or methylated states. And these patterns were consistent across diagnosis and between cell types as well. To assess the differences in methylation across diagnosis in an unbiased way, we perform a differential methylation analysis across the genome. We identify more than 200,000 differentially methylated cytosines, or DMCs, in the conventional T cells, and more than 30,000 DMCs in the Treg cell samples. In both comparisons, alveolar T cells from patients with non-pneumonia respiratory failures, that's showing red over here and blue in the Treg groups, they are relatively hypomethylated compared to those from patients with severe pneumonia. And the patients from the two pneumonia cohorts, that's severe SARS-CoV-2 pneumonia and severe bacterial pneumonia, they have a more similar methylation profile in general. Currently, we are working on identifying coordinated regulatory pathways associated with cell type, severe pneumonia etiology, and clinical outcomes by interacting our REM-seq datasets with our transcriptomic datasets. Okay. Here is a quick summary of what I talk about here today. So we show that our EM-seq demonstrated comparable performance in global CpG detections when compared to the current gold standard RBS. And where our EM-seq really shine is when profiling those CG-rich regions, especially in high coverage profiling. We also show that our EM-seq will give you a consistent performance when using ultra-low input clinical samples, and that allow us to perform methylation profiling using clinical samples from patients with severe pneumonia. Okay. Here, I want to give a huge shout out to all the members from Singer Lab for their contribution to these projects. I wouldn't be able to finish it without their contribution for sure. And also my friend and colleague, Dr. Nick Markov, for his contribution in the transcriptional analysis of these clinical samples, and also my thesis committees for all of their support. And here I also listed my programs, my department, and my funding source, and I will take any questions you guys may have. Yeah.
Thank you very much for a fascinating talk, Iris. Yeah, so we will open this for questions. If any of you have any questions, please unmute yourself and ask a question to Iris directly. Or if you prefer typing it out, please feel free to type it out on the chat and I'll read them out. There is. I can go with my first question if there is no one asking.
Okay. Arun, I think you mute yourself.
I'm sorry.
It's okay.
Yeah, okay. So you mentioned the RREM performs really well under 2 nanogram DNA samples, while RRBS. So I'm just interested in the economy of this. Like, when should the researcher choose which one? What's the most cost-effective way to deal with, like, if they have the similar number of cells and everything else is similar?
Yeah, so our usual rationale is RBS will be the cheaper way to do methylation sequencing. But it wouldn't be able to handle input usually lower than 10 nanogram. So if all you have is less than 10 nanogram, RBS wouldn't stand a chance. So you will have to use other methods, like for example, REM-seq, if you want to do your DNA methylation profile.
All right. Any questions? I can go with another one. Did you adapt your workflow from like NF Core, the BSC Bismarck or something like that, or did you write your own workflow for the analysis?
Yeah, Bismarck actually works pretty well for REM because because the cellulosine conversion rationale is the same. So anything that worked for RBS would theoretically work for REMs as well. Yeah.
Yeah, I really like the NF course methyl-seq workflow. I was just curious, like how hard it is to use everything for the workflow that you did. Yeah, good.
Well, thank you guys. Thank you, Aaron and Ali, for you guys putting this platform together. It's really great.
Can you repeat the question, please, Gabe?
It was not really a question. I was just saying thank you guys for putting this platform together. The Midwest Bioinformatics.
We thank all the speakers who volunteered to give the fantastic talk. So,
Yes, it is. It is really a success. Yeah. So thank you guys.
I think it's all the questions we have so far. Yeah, thank you again for a really compelling talk, and thank you everyone for joining us. Quick reminder: next week's speaker is Abhishek Wai from Purdue University, and yeah, so we'll post the recording for this talk on our website. Please feel free to share with your colleagues. Yeah, thank you, everyone. Thank you, Alice.
Thank you all. Thanks, everyone. Bye.
Bye.